I'm very happy I can present today on the virtual hub of Euro-BioImaging. My name is Anna Paszulewicz, and I'm the product manager for Imaris, the 3D image analysis software. Today, I would like to focus on some small parts of Imaris, being the spatial plot and the time synchronized plot, because they are very important to understand your data and to understand what is inside. So very often, spatial distribution of biological objects and the relative spatial distribution between two biological objects is related to their function and can show some interactions. So this is an important part. And another one, the time plots synchronized to the event happening in the biological system is an also important one, because often you want to measure some parameter, for example, what happens after cell mitosis, but the mitosis event doesn't happen in the same time in the cell culture. So this is where the image analysis software comes into the play and can synchronize these events for you. So I will show you a few examples and you'll learn how these tools can be used for image analysis. Okay, I think I need to. Can you see the panel on the right, or is it?
No,
okay.
We see your slides perfectly.
Right, okay. Segmentation is basically, you can call it just the first step of data analysis. So we all know there are different ways of segmentation, including machine learning and intensity-based methods. So in Imaris, we use a lot of intensity-based and machine learning pixel classification methods, which lead you to get a proper, robust, and reliable model of different structures. So there are different models for different structures. To each of them, you can get pretty easily. On the left side, you can see the retinal ganglion cell in cyan, which has been detected. So you can see the structure detected on the right side. In the same image, you can see small amacrine cells marked in magenta, which has been detected also by Imaris, and you can see the spots model on the right side. So you can see this is just the object detection, but what you can actually learn from the data. So here, the data looks very random. The researchers have been interested if there is any connection between the distribution of amacrine cell and retinal ganglion cell. So you can see now the raw data on the top, the two detected objects in the bottom. And once you split the objects, you can see there's basically no clue. So the data itself by visual inspection doesn't give you any clue if these two objects are related to one another. So the real question is, what is the truth? Are the amacrine cells located more preferably near the retinal ganglion cell? Imaris gives you the possibility to answer this question very easily using the spatial plot. So here on the plot, I will briefly present you what is on the axis. So here on the plot, you see the density of the amacrine cells on the y-axis plotted against the distance from the surface of retinal ganglion cell. So we can already see that the amacrine cells are more preferably located on the edge and close to the retinal ganglion, and their density drops while we go away from the retinal ganglion. But this is still just an observation, and all scientists need to compare this situation to some just random distribution. So in the same time, so here you can see this is the distribution of our observed amacrine cells, but in the same time, Imaris presents you the distribution, the total random distribution of spots around the retinal ganglion. So we can see, now you can compare the distribution of the real amacrine cells to the just complete random distribution. Imaris also marks you the attraction distance, so the distance up to which the probability of and the density of real amacrine cells is higher than just predicted by a complete randomness. So based on this plot, researchers can already conclude that amacrine cells, the magenta spots, accumulate preferentially around the retinal ganglion within a band of around 17 micrometer thickness. So this is the distance marked with 17 micrometers. This is the spatial plot example, but now I would like to move towards the time-lapse data example. So in this movie, you can see tumor cells attacked by neutrophils. And in the yellow channel, you can see the caspase activity. So the more yellow, the more caspase activity is observed, which indicates apoptosis. So here the question was: is touching of neutrophils? So is the connection of neutrophils to the cancer cell related to the caspase activity, and towards does it actually trigger the apoptosis event? So you can clearly see that this event, so the connection of neutrophils, touching the neutrophils to the cancer cells, happens in different points in time for every cancer structure. So this is where it's important to actually synchronize the measurements. So in this example, I synchronized all the measurements to the event which is attaching neutrophils to the cancer cell. So in this plot, on the time axis, you can see that the 0 time point means the moment when neutrophils attach to the cancer cells. And on the y-axis, you can see the intensity of caspase plotted against time, but still the 0 time point means the moment when the neutrophils attach to the cancer cells. So you can clearly see that the caspase activity grows dramatically after the attachment of neutrophils, which can already lead to the conclusion that the apoptosis measured by the caspase-7 activity here starts after the contact of neutrophils with the cancer cells. Another example related to time plots, but here not synchronized with the events, has been used within the study where researchers try to analyze the migration patterns of cytotoxic T lymphocytes to the tumoroids. So here in these two movies, you can see, so the difference between these two movies is that on the top, in the top panel, you can see the the non-cognate tumoroid on the left side. So this magenta tumoroid is not cognate. This is the naive tumoroid. And you can see the cognate tumoroid on the in the lower panel. These little green objects detected as spots in Imaris are cytotoxic T lymphocytes, and you can see their swarming movement in both panels. So just by visual inspection, you can't really tell what is going on in these data sets. They look pretty much the same. Maybe you can see a little bit more like lymphocytes here in the lower panel. But here, Imaris enables you to analyze various parameters and represent them graphically. So you can see here that each spot has a little tail, which marks the track. So you can see the tails in colors. And now you can see that these colors have a meaning. So they mean the displacement in the X direction. So displacement towards the tumoroid is here marked with minus numbers and is represented by some more reddish color, and displacement to the right, so from the tumoroid, is marked with more whiter color. So already by now, if you know the color coding for the tails, so for the tracks, you can already say, okay, so there is a little bit more movement towards the cognate tumoroid, tumorid than non-cognate tumorid. So here the tails are a little bit more red than on the top panel. So this is a very important observation, but one would like to have this observation also represented on the plot. So Imaris also enables you to create a time plot where here the displacement in X direction is plotted against time. So in the green, as in the previous example, you can see the cognate tumoroid. So this is the lower panel. And in the red color, you can see the non-cognate tumoroid. So you can see that the displacement is moved more towards the minus, so more towards the tumoroid for the cognate one as compared to a non-cognate tumoroid. So this plot already gives you an answer to the question and enables you to form a conclusion that swarming of cytotoxic T lymphocytes is directed towards cognate tumoroid as compared to the non-cognate one. So all this data is derived from the image analysis software, which is not only the plotting. So Imaris has a lot of tools, and it's a really good and complex solution for various microscopy images. So it does not only visualize your data in multiple ways, which is sort of a standard in today's microscopy. So visualization is just the first part, although it's done really nicely inside the software. The next part is the detection. So Imaris enables you to detect various models within different, using different methods, including machine learning, pixel classification, and models dedicated for the structures such as filaments or vessels. But also, the software enables you to analyze all the parameters of the detected objects and classify them. So you can classify objects for being inside or outside the structure. You can classify the objects for changing the intensity, which also enables you to create the events of interest in the time-lapse data. The classification is, of course, nicely done using machine learning classification method, which enables you to include a lot of parameters you don't even think are important, but you can mark and you can brush the objects you need on the image, and the software calculates their the classes for the rest of the objects. And the last and very important step I focused on today is the actual interpretation of the results. So not only the segmentation, which, as I mentioned, is just the first step, but also getting the reliable and conclusive information from the data ready to be published or compared with another samples. Not to mention that all those steps are batchable. So you can perform this analysis on multiple images using the same parameters, and where machine learning, of course, helps in terms of repeatability of the protocol, and is not so sensitive to changing the for the changes within the image, which all can lead to compare multiple samples between one another. So thank you very much. This is the end of my presentation for today, and I will be very happy to get some questions from you.